Hi and welcome to Optimum TV. I'm Stephen Adams and today I'm joined by Parker Moss, Programme Director of Wellcome Leap's R3 Global Programme. This initiative sets out to reinvent the manufacturing of biological medicines, particularly RNA-based medicines, to enable faster, lower cost and more scalable production. So hi Parker, thanks for joining us.
It's a real pleasure to be here, Stephen.
Great. Well, it's been 5 years or so now since the COVID pandemic, which obviously catapulted mRNA-based therapeutics into the spotlight, into the public consciousness as a new biotechnology with huge potential. But for a variety of reasons, some political, some practical, mRNA and RNA-based medicines have been something of a slow burn. Tell us about their development and what's been happening over the last 5 years.
Thanks, Stephen. Well, yes, certainly mRNA got into the consciousness of the public in the 2020s for the reason you say, but like in all areas of science, we stand on the shoulders of giants. And really the seminal work in this field was done by Katalin Kariko and Drew Wiseman in the 90s. They were the first to show that mRNA could be used as a drug. And I have to mention also Peter Cullis, who showed very cleverly in the early noughties how you could encapsulate that mRNA into a lipid and get it safely through the blood system to people's cells. So there was about 30 years of work running up to the COVID pandemic before we were finally able to optimize this incredible new and natural mechanism to vaccinate patients worldwide. And the mechanism it uses is to get the patient's own cells to produce, in this case, the spike protein, which would train the immune system to kill an incoming potential COVID virus. So that was absolutely groundbreaking, and it saved many, many millions of lives around the world. But absolutely, as you say, we've had a bit of a lull since then. There have been one or two new drug releases since Pfizer and BioNTech came in with their landmark COVID vaccines in 2021. But we haven't quite had the ramp up of new drug releases that we would have expected. And actually, that's what my organization focused on. We looked at reasons for why that has happened and what we could do to kind of re-accelerate the enthusiasm and the momentum behind mRNA. But I can tell you now from within the industry that we are about to see an absolute explosion of exuberance and enthusiasm for this modality. Because in the last few months, there have been major, major breakthroughs in cancer and actually in immunology. And these breakthroughs are ideally suited for mRNA. And I'm thinking in particular of personalized cancer vaccines, which can be mRNA encoded, but also the introduction of in vivo CAR T, which is taking a classical CAR T and having the cells manufacture that essentially in vivo inside the body. Huge implications for cancer, but also for immunological disorders like lupus.
So it's great to hear there's new excitement about RNA and mRNA-based therapeutics, and it's going to be amazing to see these new developments in the coming years. But let's talk about Wellcome Leap, the organisation you work for. Lots of people have heard of the Wellcome Trust, but not necessarily Wellcome Leap.
Yeah, I'd love to tell you about that. So I work for Wellcome Leap today. Wellcome Leap is a global DARPA-inspired health innovation organisation, and it was created by the Wellcome Trust here in London. So the Wellcome Trust are our sole shareholders, and its purpose is to pursue really high risk but also high reward scientific programs that should deliver breakthroughs in human health. And the idea is to do this in years rather than decades. So it's very, very ambitious. It has this tried and tested but really powerful operating model that came out of DARPA. And it works with seriously ambitious milestone-driven programs. So my program is called R3 Global, which I'll tell you about. And the way that we work is we pull together multidisciplinary teams. They're surge teams that come together for a short period of time, for a couple of years. We work with very aggressive timelines. We very rapidly contract experts from academia, but also from the private sector from around the world. And we do work in a globally distributed network. So my team have people on it from Singapore, from Japan, all across America, from multiple locations in the UK and across Europe as well. And the idea is that we focus on programs that have both significant scientific innovation, but a very, very practical use case. And in our case, always use cases that will impact and improve human health. And I'd love to tell you a little bit more about my very specific program, which, as you said before, is in the field of mRNA.
Okay, so the overall ethos is move fast and make things, you might say.
That's a great way of putting it.
R3 Global Program, that's one of LEAP's programs, is it? Tell us about the overall aims of that and the challenges that it's designed to overcome.
That's right. We've got about 11 programs today. Most of them are funded to between $50 and $60 million. My program has just over double that amount because it's in its second phase. And it was one of the first programs we kicked off in early 2021 in the height of COVID. And actually, it was inspired by that question you asked me right at the start, which is why, if mRNA is such a kind of promising programmable scaffold for delivering proteins to kind of humans, why has it not accelerated faster? Why don't we have 20, 30 drug approvals by now? So we really studied that question very hard, and we identified a few barriers that were just holding the industry back, and we decided to absolutely laser focus on those barriers. And I'd love to tell you about perhaps two of those to begin with. The first one was really to do with the design of protein. And it turns out that protein designers all around the world are real experts in DNA. They really have understood, thanks to sequencing, the kind of amino acid sequences of proteins, and they've become increasingly expert in protein themselves. But what most protein designers don't know much about is the very kind of esoteric field of RNA chemistry. This is things like codon optimization, secondary structures. And we did a lot of work with developing proprietary oligo libraries and with neural networks to help predict what RNA chemistry would optimize the design of proteins. So we essentially abstracted the design process and made it much easier from the manufacturing process. That was one of the major barriers that we tried to eliminate, that we think will really just reduce the barriers for protein designers to choose mRNA as their substrate. The second thing we focused on, and really where we put most of our investment and time and energy, is in the manufacturing itself. And it turns out that the tried and tested way of manufacturing any protein today is using a bioreactor, which is essentially a steel cathedral. It's a big cookery pot where you pour a recipe of reagents and enzymes and buffer in, and you literally stir the pot under pressure and heat. And after a couple of days, the proteins are produced. And there are some huge inefficiencies of doing that, mostly associated with the scale up as you go from smaller pots of protein for mouse studies to the huge building size bioreactors required to make the volumes you need for managing a pandemic. And we sought to use a very novel area of chemistry called flow chemistry, which would enable us to make proteins without the need for a bioreactor. So we funded the invention of a technology that manufactured RNA in what we call continuous flow. So we're using microfluidic flow chemistry to manufacture RNA with continuous flow, which means that there's seamless scale up using exactly the same technology that deploys plastic consumable flow cells. We can make, with the same formulation, a milligram of mRNA formulated GMP quality for a mouse, and we just run the machine for a few minutes to achieve that, all the way up to 50 grams of mRNA a day, which is enough to vaccinate a whole country. And so that's what we've sought to achieve over the last several years, and it is now working, and we've already tested that in animals, and by next year we hope to be testing this formulation in humans.
Just briefly, tell us in the simplest terms what the advantages are for those who need this, be that drug developers or whatever. Cost, I'm presuming, is it cheaper?
Yeah, so we're a charity. We're looking to impact human health, and we thought that the greatest impact we could have is making sure that the academics and young biotechs around the world who are doing successful target biology and successful studies in animal models can actually afford to then translate that into humans with affordable, high-quality clinical trial material. So this new technology will bring down the cost dramatically. It will also significantly increase the turnaround time of mRNA. We think we can get drug developers clinical trial grade, GMP grade mRNA in weeks rather than months. And importantly, it also significantly increases the quality of the drug that researchers use in mouse models. So this is really the triple whammy. We're improving the time, the turnaround time, the cost, and the quality of the drug. So it is a very significant breakthrough for anyone who wants to design proteins for both vaccines and also for therapeutics.
Fantastic. I think we've covered most of the areas we wanted to talk about, but I understand R3 Global is building a network of these RNA biofoundries around the world. Is that what's happening? Tell us about geographical expansion, distribution, etc.
Yeah, that's right. We think there are some important, maybe you could describe it as like a dual use application for our technologies. So during pandemic time, this is the ideal rapid response technology to help nation states prepare their countries or their regions for pandemic or epidemic breakouts. So we're working with governments around the world, and I'm very proud to say we're working with the British government, and we're also working with the Singapore government to deploy our technology in local biofoundries so that the moment a pandemic does arrive, those countries can be ready to manufacture rapid, high quality, very scalable mRNA-based vaccines to deal with any local or regional outbreak. But then during peacetime, which of course we hope is most of the time, that very same technology can be used to deliver vaccines, but also therapeutics for these exciting new modalities that I was talking about, where we think this will be most relevant in cancer and immunology, but also in cardiometabolic disease and neurology. And the advantage here of the kind of scale independence of our technology is that we can produce the huge volumes required for a vaccine, but also the very small volumes designed not only for a mouse study, but for an individualized medicine for a single patient, which is exactly the technology required for these new modalities that I started off talking about at the very beginning, personalized cancer vaccines and in vivo CAR T therapy.
Fantastic. Well, it's going to be fascinating to hear how this field develops in the next couple of years. Parker Moss, Program Director of Wellcome Leap's R3 Global Program. Thanks very much indeed.
It's a pleasure, Stephen. Thanks for having me on.
Bye-bye.